I'll be presenting on the SHSY5Y human cell line. This is a neuroblastoma-derived cell line that has become one of the most widely used models in neuroscience research. We'll cover its identity, its origin and features, and see how it's maintained in culture, its research applications and limitations, and finally we will discuss a primary research paper that puts all of this into context. So, the story of SHSY5Y starts in 1970 when a 4-year-old female patient was diagnosed with neuroblastoma, which is a cancer that arises from immature nerve cells. Then, a metastatic bone marrow biopsy from this patient was used to establish the original parental cell line SKNSH cell line. From that, the parental cell line, a subline was derived and then cloned thrice, which is where the SY5Y came from. Reflecting the successful cloning history, the result is what we have now, the SHSY5Y cell line. Today, it is commercially available through ATCC under catalog number CRL2266. Because these cells are genetically identical clones, they offer a level of experimental consistency that you simply cannot get from other primary neurons, which varies from donor to donor and batch to batch. This reproducibility is a huge part of why this line became so popular. So now let's look at what makes this line biologically distinctive. Genetically, the cells have a mode chromosome number 47, which is one more than the normal. The unique chromosomal marker is a partial trisomy of a long arm of chromosome 1. More, they lack a strong amplification of the MYCN oncogene, which is the hallmark of aggressive neuroblastomas. So this makes them more stable and manageable for research purposes. In terms of the enzymatic profile, SHSY5Y cells express two key enzymes in their catecholamine pathway. First, tyrosine hydroxylase, which is the rate limiting enzyme in dopamine synthesis. Second, dopamine beta hydroxylase, which converts dopamine into non-adrenaline. They also express dopamine D2 and D3 receptors, which makes it relevant for studying movement disorders. And when differentiated, they upregulate mature neuronal markers like beta 3 tubulin, synaptophysin, and PSD95, all of which give confirmation of mature neuronal identity. So, to culture these cells, SHSY5Y are maintained in a 1:1 mixture of EMEM and F12, supplemented with 10 to 15% fetal bovine serum, depending on the supply's recommendation. They kept at 37 degrees Celsius in a humidified incubator with 5% CO2, and they prefer slightly acidic conditions. For experiments involving neurite growth, a specialized surface coating called Geltrex is required. One thing that makes subculturing these cells a bit more complex than a typical adherent line is their mixed morphology. They grow as they grow as a mixed population with both floating neuroblastic clusters and adherent epithelial-like cells. So the protocol requires you to first collect those floating cells from the spent media, then trypsinize the remaining adherent population, combine both the fractions and replace at a 1:20 or 1:50 ratio. The doubling time is approximately 48 hours. Now, when we are working with this cell line, we need to identify whether they are differentiated or not, because this is essentially they become two different research tools. Undifferentiated cells have a mixed morphology. They're only immature neuronal markers and lack a true dopaminergic phenotype. They're useful for broad neurotoxicity assays, high throughput screening. However, they are not appropriate for tau related research because tau phosphorylation levels fluctuate dramatically during the differentiation process, so results would be like unreliable. Uh, Differentiated cells are typically achieved by treatment with retinoic acid and followed by brain-derived neurotropic factors. They develop extensive neurite growth and branching. They express mature dopaminergic markers, which we talked about in slide 3, and they are used for highly specific disease models like Parkinson's disease. Tau phosphorylation also stabilizes in this state, and so they are appropriate for that line of research. So now we compare why we could choose the SHSY5Y over other alternatives like choosing rodents or iPSCs. The main benefit that the SHSY5Y comes with is scalability and cost. Primary neurons are difficult to isolate, expensive, and vary greatly between preparations. Human iPSC derived neurons are the gold standard for physiological relevance, but they come with a huge cost, complexity, and long differentiation timelines. One important caveat for the SHSY5Y is the Warburg effect. Because these cells come from a tumor, their energy metabolism shifts a lot towards glycolysis, even in the presence of oxygen. This means that the baseline mitochondrial function differ from healthy neurons, which can alter our interpretation on certain experiments. But this makes our cell line very useful for early stage experiments. And once done, the researcher can shift to rodents or iPSs to validate the results. So, with that in mind about SHSY5Y, I want to walk through a primary research paper that I found, which which puts this cell line in practice. The paper is by Rizal et al. 2021, published in PNJ, and the title is Icaritin protects SHSY5Y cells transfected with TB43 by elevating mitochondrial damage and oxidative stress. That's a lot. The main question that the paper addressed was, can the natural compound Icaritin protect neuronal cells against the damage caused by abnormal expression of TDP43? So a little about TDP43. Under normal conditions, TDP43 is a nuclear protein. It involves in RNA splicing, stability, and transport. But when it is overexpressed or mislocalized to the cytoplasm, as it happens in diseases like ALS or dementia, it accumulates in the mitochondria, disrupts the respiratory chain, reduces ATP production, and generates oxidative stress. This makes it a clinically relevant disease target. The experiment had three stages. First, the researchers cultured SHSY5Y cells in a DMEM F12 medium and transfected them with a viral vector to overexpress the TDP43. Then they optimized the viral multiplicity of infection, finding that an MOI of 30 for 12 hours gave effective mitochondrial damage and oxidative stress without killing the cells. This was also confirmed by a fluorescent microscopy. Second, they administered the Icaritin at varying concentrations, zero point 0.1, 1, and 10 micromolar across different time points. They found that 1 micromolar for 48 hours gave optimal cell viability. Third, they use three main readouts: the CCK assay to measure the cell viability, the JC fluorescent probe to assess the mitochondrial membrane potential, where the red fluorescent showed healthy mitochondria and green showed compromised ones, and Western blot to quantify the TDP43 and cytochrome C expressions. They also measured ATP content and antioxidant activity. And the results were very clear across the experiment. Um, when you see the control group, it's the fluorescent microscopy shows red, which shows that the mitochondria is healthy. Control plus iCandidate also shows red, which means the drug did not affect the mitochondria adversely or showed any other effects. And TDP43 overexpression, we see green, which means that the mitochondrial memory potential was affected. And when we see the TDP43 treated with Icaritin, we see red or yellowish tint, which shows that it's working and it's doing the treatment. Um, we see the statistical analysis. We see that TD43 has a lower red-green ratio, which we saw in the image, and with icaritin treatment, it increases. ATP production is less in TDP43 overexpressed cells, but when they're treated with icaritin, it again goes back, almost reaches normal. uh, uh, cytochrome C cytochrome C expression, when the membrane potential of mitochondria break, cytochrome C is released, so it's expressed more, and that's what we see in TDP43 overexpressed cells. But when you go to the TDP43 icaritin treated cells, we see that it again goes down. but not significantly, but it still goes down. So the treatment works. Putting all of this together, the proposed mechanism of Icaritin gives four actions. It downregulates the TDP43 expression, stabilizes the mitochondrial membrane potential, and increases ATP content and boosts antioxidant enzyme activity. The authors shows that Icaritin promotes TDP downregulation through an ATP-dependent mitochondrial protease called LON-T1, but this was not directly measured. So finally, to conclude, Icaritin successfully rescued the function and ADP production in TDP43 transfected SHSY5Y cells. This shows primary evidence of neuroprotective potential in a possible neurodegenerative models, a disease model like ALS or Parkinson's. To circle back, why SHSY5Y was used? Because this is a human-derived neuronal model that can replicate the oxidative stress and mitochondrial dysfunction similar in ALS type models. It was scalable, it was reproducible, cheap, and just something that we wouldn't have achieved with primary neurons. So, thank you for watching my presentation.